Retinal cell biology. Exposure to blue light reduces melanopsin expression in intrinsically photoreceptive retinal ganglion cells and damages the inner retina in rats. Natalia, Ziolkowska, 1. Malgorzata, Chmielewska, Kresinska, 2. Ala-Winiarska, 3. and Waldemar Sienkiewicz, 4. 1. Department of Histology and Embryology, Faculty of Veterinary Medicine, University of Warmia and Masaryk, in Olsztyn, Poland. 2. Department of Pathophysiology, Forensic Veterinary and Administration, Faculty of Veterinary Medicine, University of Warmia and Masaryk, in Olsztyn, Poland. 3. Department of Pharmacology and Toxicology, Faculty of Veterinary Medicine, Stepan Gzhitsky National University of Veterinary and Biotechnologies, Lviv, Ukraine. 4. Department of Animal Anatomy, Faculty of Veterinary Medicine, University of Warmia and Masaryk, in Olsztyn, Poland. Correspondence: Natalia, Ziolkowska, Department of Histology and Embryology, Faculty of Veterinary Medicine, University of Warmia and Masaryk, in Olsztyn. Oksapowskiego, 13 Str. 10 to 693, Olsztyn, Poland. Natalia.Terezaska at uwm.edu.pl received September 9, 2021. Accepted December 24, 2021. Published January 21, 2022. Citation: Ziolkowska N., Chmielewska Kresinska M., Winiarska A., Sienkiewicz W. Exposure to blue light reduces melanopsin expression in intrinsically photoreceptive retinal ganglion cells and damages the inner retina in rats. Invest Ophthalmal Visci, 2022-631, doi.org/10.167/IOVs.63.126. The purpose of this study was to investigate the effects of blue light on intrinsically photoreceptive retinal ganglion cells, iPRGCs. Methods: brown Norway rats were used. 9 rats were continuously exposed to blue light, light-emitting diodes, LDs, 463 nanometers, 1000 LX. For 2 days. Acute exposure, AE. 9 rats were exposed to 12 hours of blue light and 12 hours of darkness for 10 days, long-term exposure, LTE. 6 control rats were exposed to 12 hours of white fluorescent light, 1000 LX, and 12 hours of darkness for 10 days. Whole mount retinas were immunolabeled with melanopsin antibodies, melanopsin positive, MP. iPRGC somas and processes were counted and measured with neuron J. To detect apoptosis, retinal cryosections were stained with terminal deoxynucleotidal transferase DO-P-NIC-end labeling. Ultra-thin sections were visualized with transmission electron microscopy. Results: the number of MP-iPRGC somas was significantly lower in retinas from A&E and LTE rats than in those from control rats, p less than 0.001 and equal to 0.002 respectively. The mean length of MP areas of processes was significantly lower in A&E rats, p less than 0.001. A&E rats had severe retinal damage and massive apoptosis in the outer nuclear layer. Their mitochondria were damaged in the axons and dendrites of the nerve fiber layer and the inner plexiform layer. Retinal ganglion cells, RGCs, in AE rats appeared to have reduced amounts of free ribosomes and rough endoplasmic reticulum. Conclusions: A&E to blue light reduces melanopsin expression and damages RGCs, likely including iPRGCs. Changes in the axons and dendrites of RGCs suggest possible disruption of intraretinal and extraretinal signal transmission. Keywords: melanopsin, rat, blue light, intrinsically photoreceptive retinal ganglion cells, retinal damage. As the use of devices emitting blue-enriched light has increased, so have concerns about its negative effects on the retina. 1. 2. Excessive exposure to blue light can cause insomnia and transient smartphone blindness, damage the retina, and threaten vision. 2.7. In general, the retina is most sensitive to shorter wavelength light, which causes damage that is primarily photochemical in nature. Blue light causes photoreceptor death and retinal gliosis, which leads to retinal degeneration. 4 to 7. 9 to 14. Blue light exposure may also affect intrinsically photoreceptive retinal ganglion cells, iPRGCs, as these cells contain melanopsin, a photopigment with peak absorbance at around 480 nanometers. The iPRGCs play roles in image-forming responses 1617 and in non-image-forming responses 1.7. In the rat retina, these neurons constitute about 1% to 3% of the RGC population and are most concentrated in the superotemporal region. 1819 decreases the number of iPRGCs in the rat retina by downregulating melanopsin expression and changes melanopsin localization within these cells. However, even though it is known that blue light-emitting diodes can be more damaging to the retina than white or green light, 451,314 the effects of monochromatic blue light exposure on rat iPRGCs have not been investigated. The iPRGCs could be particularly susceptible to blue light irradiation because their axons are unshielded by myelin and laden with mitochondria. Mitochondrial enzymes are affected by blue light, which can lead to generation of. This work is licensed under a Creative Commons Attribution 4 international license. 1. Blue light reduces melanopsin expression, IOVs, or January 2022 Volley 63 or NOBA 1, or Article 26 or 2, reactive oxygen species and affect axonal transport and the survival of iPRGCs, 24 to 26. Moreover, the only studies involving long-term exposure to blue light have investigated its effects on photoreceptors, 9.27, not iPRGCs. For this reason, more long-term studies on animal models are needed. Additionally, in studies on the effects of exposure to high-intensity white light on iPRGCs, only albino rats were used. However, light-induced retinal degeneration differs in pigmented and albino rats, as does regulation of melanopsin mRNA and protein expression. As does response of iPRGCs to light exposure may also differ between these strains, but studies on pigmented rats are needed to provide a basis for comparison. Therefore, our objective was to determine the effects of acute and long-term exposure to blue light, 1000 LX, on the retinas of pigmented rats, with a particular focus on iPRGCs. To examine and count iPRGCs in whole-mount retinas, we used immunolabeling for melanopsin. For measuring the length of iPRGC processes, we used NeuronJ. We supplemented these techniques with electron microscopy, and for labeling apoptotic cells, we used terminal deoxynucleotidyl transferase DUTP nickend labeling, TUNEL. Methods: animals and experimental design. 3-month-old male and female brown Norway rats, Charles River laboratories, were used. All rats were kept in 12 hours of white fluorescent light and 12 hours of darkness for the first 3 months of life. Fluorescent lamps provided white light, less than or equal to 100 LX. Then, the acute exposure, A&E, group 9 rats, was first kept in total darkness for 24 hours, and then continuously exposed to blue light for 48 hours. The long-term exposure, LTE, group 9 rats, was exposed to 12 hours of blue light and 12 hours of darkness for 10 days. The control group, 6 rats, was kept in 12 hours of white light, 1000 LX, and 12 hours of darkness for 10 days. To dilate the rats' pupils in all 3 groups, 1. Atropine drops were applied twice a day. All rats were kept in transparent plexiglass cages, supplementary Fig. S1. Blue light was provided by clusters of light-emitting diodes, LEDs, 463. 10 nanometers, 400 blue diodes per cage. 50 diodes provide 16,000 lumen, 5 meters to. White and blue LED illumination was measured at the level of the rat's eyes with a monochromatic light meter, multi-LED 10 Mars. Taiwan. Supplementary Figure S1 shows other details of the experimental system. All rats were euthanized at 8 a.m. in a CO2 chamber. All experimental procedures were performed according to Ukrainian animal welfare regulations and were approved by the Poltava National Agricultural Academy Ethical Commission. Rat retina whole mounts were prepared as in Dreising et al. 31 briefly. The eyes were enucleated immediately after euthanasia. Next, the retina was gently dissected from the eye cup and fixed in 4% paraformaldehyde for 45 minutes, then washed in PBS. For immunocytochemistry, the tissues were incubated for 48 hours with an antibody against melanopsin, 1/20 dilution, ABCAM 19,383, in 0.1 millions phosphate buffer, containing 2% BSA and 0.1% tween. After 3 15-minute rinses in 0.1 millions phosphate buffer, the tissues were incubated with secondary antibody, 1/20 dilution, Alexa 568, in 0.1. Immunostaining. M-phosphate buffer for 3 hours. Retinas were washed with PBS, then mounted in Gelvatol mounting medium containing Dabco, Sigma, Germany, and Floramount, G32. Counting of IPRGC somas and determination of melanopsin-positive neural process lengths. Melanopsin-positive IPRGC somas in entire whole mounts were counted manually with image J151N software 33. To determine neural process lengths, 4 images from the temporal retina were randomly selected from each retina and opposed 1.28 Hoffi's MM2 composite image. The same was done with 4 images from the nasal retina for a total area of 2.560 mm2. Melanopsin immunoreactive nerve fibers were traced manually with neuron J34% for image J, and the length of all the fibers in the 2.560 mm2 area was summed. The IPRGC somas numbers in this area were also counted manually with image J tunal assay. For apoptosis detection, a tunal assay was performed using an in situ cell death detection kit, Roche Diagnostics GmbH Mannheim, Germany, according to the manufacturer's instructions. 8 cross-sections of each retina were examined. Supplementary table S1. Tunel-positive nuclei were counted manually in a 1,000 micrometers long area of each section. In cases where nuclei may have fragmented, each potential fragment was counted as an individual nucleus. Electron microscopy. For electron microscopy, the dorsal half of retinas was collected within 3 minutes of heart stoppage. See supplementary table S1. Tissue pieces were fixed 2 hours, 4 degrees Celsius, in a mixture of 1% paraformaldehyde and 2.5% glutaraldehyde in 0.2 millions phosphate buffer, pH 7.4. Samples were washed, post-fixed in 2% osmium tetroxide, 2 hours at room temperature, and embedded in EPON 812. Semi-thin sections were stained with toluidine blue and examined to identify regions for electron microscopy. Ultra-thin sections were contrasted with urinal acetate and lead citrate, then examined using a Techni 12 Spirit G 2 biotwin transmission electron microscope. TEM, FE, USA. 5 to 7 cross-sections of each retina were examined by TEM. Statistical analyses. Differences in IPRGC soma numbers and melanopsin-positive neural process lengths were analyzed with Tukey's HSD. R4.435, sensitivity analysis with trimmed means 36, indicated that the results were minimally affected by potential deviations from normality and heteroscedasticity. Results. Exposure to blue light reduced the number of melanopsin-positive IPRGC somas in rat retinas. The A&E and LTE groups had significantly fewer melanopsin-positive IPRGC somas in the retina than the control group. Figure 1. Although the AE group had significantly fewer of these somas in the retina than the LTE group, the 95% confidence interval, CI, for the difference included values near 0. Similar results were observed in the smaller 2.56 VRM&U. Blue light reduces melanopsin expression. IOVs, or January 2022, or Vol. No one, or Article 26. Figure 1. Blue light exposure reduced the number of intrinsically photoreceptive retinal ganglion cells, iPRGCs. Brown Norway rats were exposed to 12 hours of white light, 1000 LX, and 12 hours of darkness for 10 days. Control, 6 rats. 12 hours blue light, 1000 LX, and 12 hours darkness for 10 days. Long-term, 9 rats, and blue light, 1000 LX for 2 days. Acute, 9 rats. Red fluorescence shows staining for melanopsin. A. Control group. B. Long-term exposure group. And C. Acute exposure group. D. Circles indicate number of iPRGCs in one complete whole-mount retina from one rat. Horizontal lines show mean. E. Dots indicate differences between groups with regard to mean counts of iPRGCs in whole-mount retinas. Error bars show 95% confidence intervals for the differences. The p-values and confidence intervals were calculated with Tukey's HSD test, R, version 4.0.4, after sensitivity analysis, indicated that deviations from the assumptions of normality and homogeneous variance would not affect conclusions about the statistical significance of differences. Area that was used for measuring neural process lengths, see below, Figure 2. The AE group had significantly fewer melanopsin-positive iPRGC somas than the LTE and control groups, and the LTE group had significantly fewer of these somas than the control group. Acute exposure to blue light changed melanopsin distribution in iPRGCS. Melanopsin distribution in iPRGCS was similar in the control and the LTE groups but differed in the AE group. The intensity of staining in the somas and processes was less intense in the A&E group. In the A&E group, immunoreactivity to melanopsin was sometimes limited to soma membranes. In most processes in the A&E group, the staining did not extend as far from the soma as it did in the other groups, see Figs. 2AF. In the A&E group, melanopsin expression was reduced mostly in the outer stratifying dendrites of the inner plexiform layer, see Figure 2F. Although melanopsin-positive varicosities were present on the processes in all three groups, they were less frequent and less prominent in the A&E group, see Figs. 2E, 2F. Acute exposure to blue light reduced the mean length of iPRGC neural processes in rat retinas. The mean length of melanopsin-positive iPRGC neural processes in the examined 2.560 mm2 areas was significantly lower in the A&E group than in the LTE and control groups, Figure 3. The mean length of these nerve fibers was similar in the LTE and control groups. Retinal damage after blue light exposure. Clear signs of retinal damage were visible in the A&E group. Some retinal abnormalities were detected in the LTE group, and no retinal abnormalities were seen in the control group. Light microscopy revealed that the photoreceptor layer had deteriorated in A&E retinas, Figure 4C, but not in LT retinas, Figure 4B. In AN retinas, the outer nuclear layer had disappeared, and only scattered nuclei were present, Figure 4C. In those scattered nuclei, indicators of apoptosis were present, for example chromatin condensation, see Figure 4C. Although the outer and inner photoreceptor segments were visible in most regions of A&E samples, compared to the other groups, they were usually less numerous and shrunken, see Figure 4C. In. Blue light reduces melanopsin expression. IOVs or January 2022 vol. 63 or number 1 or Article 26. 4. Figure 2. Effect of blue light exposure on melanopsin distribution in intrinsically photoreceptive retinal ganglion cells, iPRGCs, in whole-mount retinas from brown Norway rats. The rats were exposed to 12 hours of white light, 1000 LX, and 12 hours of darkness for 10 days. Control, 6 rats, 12 hours of blue light, 1000 LX, and 12 hours of darkness for 10 days. Long-term, 9 rats, and blue light, 1000 LX, for 2 days. Acute, 9 rats. A. Control group, 20X. Note, dense network of melanopsin-positive dendrites. B. Control group, 40X. Multiple melanopsin-positive processes with varicosities are visible. C. Long-term exposure group, 20X. A widespread dendritic network of melanopsin-positive processes is visible. Note the smaller number of melanopsin-positive iPRGC somas. D. Long-term exposure group, 40X. Multiple melanopsin-positive varicosities are also visible here. Note, 2 melanopsin immunoreactive cells located in the ganglion cell layer, arrow, and in the inner nuclear layer, double arrow. E. Acute exposure group, 20X. Note, levels of melanopsin expression differ widely in the iPRGC somas. Relative to the other groups, melanopsin immunoreactivity is decreased in the dendrites, and the melanopsin-positive varicosities on the nerve fibers are less prominent. F. Acute exposure group, 40X. Melanopsin is present mostly in iPRGC somas and proximal dendrites. Note: lack of varicosities on these processes and decreased melanopsin immunoreactivity in outer stratifying dendrites, arrows. Samples were visualized with a confocal microscope, LSM 700, Zeiss, G. Circles indicate number of iPRGCs in a 2.5 per 6, 0 mm2 area of one retina from one rat. Horizontal lines show mean. E. Dots indicate differences between groups with regard to mean counts of iPRGCs. Error bars show 95% confidence intervals for the differences. The AE group, signs of apoptosis were occasionally present, for example chromatin condensation and marginalization. See Figure 4C. Control and LTE samples in contrast, the outer and inner segments had normal morphology. In the A&E group, but not in the control and LTE groups, see Figs 4A, 4B, the inner plexiform layer and the nerve fiber layer were thick and vacuolized. See Figure 4C. In the inner nuclear layers, INLs, in electron micrographs, the outer nuclear layer looked normal in the LTE and control groups. Figs 5A, 5B, in blue light reduces melanopsin expression. IOVs, or January 2022, or Volley. No one, or Article 26, 5, Figure 3. Acute exposure to blue light reduced the mean length of processes of intrinsically photoreceptive retinal ganglion cells, iPRGCs. Brown Norway rats were exposed to 12 hours of white light, 1000 LX, and 12 hours of darkness for 10 days. Control, 6 rats, 12 hours of blue light, 1000 LX, and 12 hours of darkness for 10 days. Long term, 9 rats, and blue light, 1000 LX, for 2 days, acute, 9 rats. A. Circles indicate total length of iPRGC processes in a 2.560 mm2 area of the retina from one rat. Horizontal line shows mean. B. Dots indicate differences between groups. Error bars show 95% confidence intervals for the differences. Figure 4. Effect of blue light exposure on retinal morphology of brown Norway rats. The rats were exposed to 12 hours of white light, 1000 LX, and 12 hours of darkness for 10 days. Control, 6 rats, 12 hours of blue light, 1000 LX, and 12 hours of darkness for 10 days. Long term, 9 rats, and blue light, 1000 LX, for 2 days. Acute, 9 rats. Resin-embedded semi-thin sections were stained with toluidine blue. Retinas from control, A, and long-term, B, exposure groups display normal morphology. C, retinas from the acute exposure group had deteriorated. Note, loss of nuclei in the outer nuclear layer. Photoreceptors are shrunken and sparser than in the other groups. Signs of apoptosis, chromatin condensation and marginalization are present in the inner nuclear layer, double arrow. The inner plexiform layer and nerve fiber layer are thicker than in the control group and display vacuolization, arrows. Note the pigment granules scattered in the outer nuclear layer, arrowheads. Samples were visualized with a confocal microscope, LSM 700. Zeiss. The A&E group, however, there were markedly fewer nuclei in this layer, and cells displayed signs of different stages of apoptosis, pycnotic nuclei containing mostly heterochromatin, vacuolized nuclei, and nuclei that had collapsed, see Figure 5C. Most of the inner segments looked normal in the LTE and control groups, see Figs. 5D, 5E. In the A&E group, most inner segments had disrupted cell membranes and swollen mitochondria. See Figure 5F. Photoreceptor outer segments were commonly disarranged in the A&E group, and sometimes blue light reduces melanopsin expression. 6. IOVs or January 2022 Vol. 63 or Number 1 or Article 26. Figure 5. Electron micrographs of retinas from brown Norway rats. The rats were exposed to 12 hours of white light, 1000 LX, and 12 hours of darkness for 10 days. Control, 6 rats, 12 hours of blue light, 1000 LX, and 12 hours of darkness for 10 days. Long-term, 9 rats, and blue light, 1000 LX, for 2 days. Acute, 9 rats. Normal-looking outer nuclear layers, ONL, in A, control, and B, long-term exposure groups. C. ONL from acute exposure group. Note: nuclei are sparser. Stages of apoptosis are evident. Star equal to pycnosis. Asterisk equal to chromatin pycnosis and vacuolization. And arrowhead equal to collapsed nuclei. And multiple processes of Muller cells are present between inner segments, arrows. Pigment granules are present between scattered nuclei, double arrow. Normal-looking photoreceptor outer and inner segments from D, control, and E, long-term exposure groups. Note: some outer segments are disarranged, arrows. F. Inner segments from acute exposure group have disrupted membranes and swollen mitochondria, arrows. Note: vacuolization of the transition zone between photoreceptor inner and outer segments, arrowheads. Ultrathin sections were visualized using a Techni 12 Spirit G2 biotwin transmission electron microscope, FE, USA, equipped with 2 digital cameras, a Velita, Olympus, Japan, and an EGE 4K, FE, USA. Disarranged in the other 2 groups. In AE retinas, most of the rod outer segments had disarranged discs, and vacuoles were present at the transition zone between the inner and outer photoreceptor segments. See Figs. 5C, 5F. Multiple Muller cell processes were present in the subretinal space of A&E rats, but not in retinas from the other rats. In A&E retinas, these processes extended between inner photoreceptor segments. See Figure 5C. In the A&E group, the nerve fiber layer and the inner plexiform layer showed signs of damage. Figs. 6, Sig, 6I. They were vacuolized, and the mitochondria in axons and dendrites were markedly swollen and had disrupted cristae. However, in the LTE and control groups, these layers looked normal. See Figs. 6A, 6 billions, 6 grams, 6 hours. Finally, ganglion cells appeared normal in control and LTE rats. See Figs. 6D, 6E. In A&E rats, these cells had swollen mitochondria and sparser ribosomes and cystins of the rough endoplasmic reticulum. See Figure 6F, Tunnel assay for apoptosis. In control retinas, only isolated apoptosis signals were detected, 2 to 6 nuclei per rat. Figure 7. Supplementary Table S2. In LTA retinas, scattered individual nuclei were labeled positive in the ONL, 12 to 27 nuclei per rat. In A&E retinas, however, massive labeling was detected in the ONL, indicating widespread apoptosis of photoreceptors. Approximately 3,878 to 5,126 nuclei per rat. Note that nuclear fragmentation made exact quantification difficult. In A&E retinas, occasional positive labeling was detected in individual nuclei in the INL. Discussion. Our results indicate that LTE and A&E to blue light reduce the number of melanopsin-positive iPRGC somas in the retinas of pigmented rats. A&E, but not LT, substantially reduces the length of melanopsin-positive iPRGC neural processes and damages photoreceptors. Whereas LTE does not appear to affect retinal ganglion cells, RGCs, AE causes lesions in the axons in the nerve fiber layer and in the dendrites in the inner plexiform layer, as well as minor changes in RGC somas. It is possible that iPRGCs may not have been detected because exposure to blue light could have reduced melanopsin expression in these cells. Thus, although we cannot be certain whether exposure to blue light reduces the number of iPRGCs or whether it downregulates melanopsin expression in these cells, our results clearly indicate that blue light reduces melanopsin expression. IOVs or January 2022 or Vol 63 Number 1 or Article 26. Figure 6. Electron micrographs of retinas from brown Norway rats. The rats were exposed to 12 hours of white light, 1000 LX, and 12 hours of darkness for 10 days. Control, 6 rats, 12 hours of blue light, 1000 LX, and 12 hours of darkness for 10 days. Long-term, 9 rats, and blue light, 1000 LX, for 2 days. Acute, 9 rats. In the A control and B long-term exposure groups, nerve fiber layer axons and their mitochondria appear normal. C. In the acute exposure group, axons are swollen and have mitochondria with disrupted cristae, arrowheads. Somas of ganglion cells from the D control and E long-term exposure groups display normal ultrastructure with numerous free ribosomes and cystins of rough endoplasmic reticulum, arrows. F. Ganglion cell somas from the acute exposure group have fewer free ribosomes and cystins of the rough endoplasmic reticulum, as well as swollen mitochondria, arrow. In retinas from the G control and H long-term exposure groups, the inner plexiform layer appears normal. I. In the retina from the acute exposure group, the inner plexiform layer is vacuolized. Ultrathin sections were visualized using a Techni 12 Spirit G2 biotwin transmission electron microscope, FE, USA, equipped with 2 digital cameras, a Velita, Olympus, Japan, and an EGE 4K, FE, USA. iPRGCs are affected by exposure to blue light. In future studies, a marker for all RGCs will be used to clarify the effect of blue light on the entire population of these cells and to elucidate differences in the response iPRGCs and other RGCs to blue light exposure. Our count of 900 to 1,500 iPRGCs in retina whole mounts from normal pigmented rats is consistent with the results of other studies. 181,937. Additionally, our observations that, in the LTE and AE groups, the number of iPRGC somas was reduced and that labeling for apoptosis was absent in the ganglion cell layer are similar to the findings of others, 20.21 who exposed albino rats to white light, 3,000 LX for 2 days or 200 LX for 2 to 8 days. Light and darkness regulate melanopsin expression in the retinas of pigmented 28 and albino rats. Hamsters and mice, extended exposure of rats to constant white light for 1 to 5 days downregulates melanopsin expression, resulting in a reduced number of iPRGC somas. Taken together, these studies and ours indicate that the reduction in the number of iPRGC somas was not caused by cell death, but it could be due to reduced blue light reduces melanopsin expression. IOVs or January 2022, Vol. 63 or Number 1 or Article 26. 8. Figure 7. Tunnel assay of brown Norway rat retinas. Blue fluorescence shows nuclei stained with DUPI. The rats were exposed to 12 hours of white light, 1000 LX, and 12 hours of darkness for 10 days. Control, 6 rats. 12 hours of blue light, 1000 LX, and 12 hours of darkness for 10 days, long-term, 9 rats, and blue light, 1000 LX, for 2 days, acute, 9 rats. A. Retina from a rat in the control group. Note, no labeling for apoptosis. B. Retina from a rat in the long-term exposure group. Isolated nuclei display labeling for apoptosis, white arrows equal to green fluorescence. In the ONL. C. Retina from a rat in the acute exposure group. Note, massive labeling for apoptosis is present in the ONL, white arrows equal to green fluorescence. Samples were visualized with a confocal microscope. LSM 700. Zeiss. Abbreviations: ONL, outer nuclear layer, INL, inner nuclear layer, GCL, ganglion cell layer, detection of somas caused by decreased melanopsin protein expression. We found that A&E to blue light reduces the total length of the melanopsin-positive areas of processes. There are at least 3 factors that could explain this finding and the reduction in the density of melanopsin-positive varicosities on those processes. First, downregulation of melanopsin expression may have played a role. Hannibal et al. 28% showed that melanopsin expression was significantly reduced after a prolonged period of constant light exposure. Moreover, melanopsin protein expression is transiently downregulated in the retina of albino rats exposed to high-intensity cool white light. 20. This downregulation is probably a protective mechanism because melanopsin produces reactive oxygen species when it is activated by blue light, 41 to 43. Second, these effects of A&E to blue light on iPRGCs could be due to changes in the distribution of melanopsin, which has been shown to change in rat retinas in response to constant illumination or constant darkness. 212,838. For example, after 8 days of exposure to 200 LX of white light, melanopsin immunoreactivity was no longer detected in iPRGC processes and was seen only in their somas. Similarly, after 2 days in constant white light at 300 LX, melanopsin immunoreactivity was decreased in the distal dendritic processes. After 3 days, it was also decreased in the proximal processes, and after 5 days, it was only visible in the somas. 38. Similar changes occurred in pigmented rats continuously exposed to white light, and the changes were more prominent in the outer stratifying processes than in the inner ones. 28, as we also observed in our rats after continuous exposure to monochromatic blue light. Hannibal et al. 28 postulated that newly synthesized melanopsin is transported from the soma to the processes, and that the soma membrane may be the last place where melanopsin expression is downregulated in response to light exposure. In our rats, the mean length of melanopsin-positive processes in an area of 2.56 mnm2 was slightly longer in the LTE group than in the control group. However, the 95% CI indicates that, in all such rats, the mean length of these areas could be reduced after LTE to blue light. Thus, our data indicate that A&E reduces the mean length of melanopsin-positive areas in iPRGC processes and leaves open the possibility that LTE could also reduce their length, but to a lesser extent. Third, these effects on iPRGCs might also be due to photoreceptor damage. However, it remains unclear whether photoreceptors play a role in regulating melanopsin expression in iPRGCs. On the one hand, some studies indicate that classical photoreceptors interact with iPRGCs 3904 to 46, form synapses with bipolar and amacrine cells, suggesting that rod and cone input may be involved in regulating melanopsin expression. In mice, the outer stratifying dendrites of iPRGCs are in close proximity to cone photoreceptor terminals, which suggests a functional connection. In transgenic mice with degenerated photoreceptors, melanopsin expression was undetectable. Additionally, 1 et al. 47 found that 28 days after rats were injected intraperitoneally with N-methyl-N-nitrosourea, their photoreceptors had degenerated and melanopsin protein expression in iPRGCs had decreased markedly, although it persisted in the soma for a long time. They suggested that intact photoreceptors may be necessary for the normal distribution of melanopsin in iPRGCs. On the other hand, some studies suggest that melanopsin expression by iPRGCs is independent of classical photoreceptor input. For example, the appearance of iPRGCs and melanopsin distribution within these cells did not differ between rodless, coneless mice and age-matched, wild-type mice. For the ACE, lacking certain enzymes that regulate the visual cycle, the melanopsin photocycle can occur independently of both the rod and cone photocycles. In neonatal albino rats, in which rods and cones are not fully developed, changes in melanopsin protein expression were independent of rod and cone input. Moreover, 1 month after albino rats were exposed to white light, 3000 LX, melanopsin expression began to recover gradually, while photoreceptors continued to die. Some of these discrepancies between studies may be due to differences in the experimental protocols and the models that were used, that is, light-induced retinal damage versus chemically induced damage versus knockout rodent models. However, it remains unclear whether the damage to the photoreceptors in our rats was related to the changes in their iPRGCs. Blue light reduces melanopsin expression. IOVs or January 2022 or Vol. No one or Article 26. Several of our observations suggest that A&E to blue light damage the iPRGCs. First, the melanopsin-positive varicosities were less numerous and less prominent in the A&E group than in the other groups. Second, in the inner plexiform layer, all dendrites were vacuolized and had swollen mitochondria with disrupted cristae. Third, in the nerve fiber layer, the same lesions were observed in all the axons. Fourth, in RGCs in the ganglion cell layer, the number of free ribosomes and of cistons of the rough endoplasmic reticulum appeared to be lower in the A&E group than in the other groups. Apoptosis signals were not detected in the ganglion cell layer, but it is possible that, if the exposure had been prolonged, these cells would have undergone apoptosis. Although iPRGCs cannot be distinguished from other RGCs by electron microscopy, all RGCs in these sections displayed these abnormalities. To identify possible differences in the degree to which RGCs and iPRGCs respond to blue light exposure, future studies could use immunoelectron microscopy. These lesions and changes in the iPRGCs may be due to several causes. RGCs are susceptible to blue light exposure because their axons and dendrites contain numerous mitochondria, 2223. These organelles can be harmed by short-wavelength blue light, which causes oxidative stress and reduces RGC cell survival, 50.51. The varicosities in iPRGCs contain large numbers of mitochondria, 23, which suggests that the mitochondrial lesions we observed could have been related to the reduced number and prominence of melanopsin-positive varicosities. The iPRGCs can be more resistant than other RGCs to some injuries like axotomy or optic nerve injury, 52 to 54, but in other models, for example experimental glaucoma or inherited photoreceptor degeneration, the iPRGC death rate is similar to that of rods and cones. 55.6. The iPRGCs seem to be more resistant to direct damage from light exposure than conventional photoreceptors, 4.6.9. However, our results indicate that A&E to blue light was probably sufficient to damage iPRGCs, and that this damage was probably the direct result of blue light exposure, as there was not enough time for remodeling processes to damage these cells. The molecular mechanism of light-induced damage to RGCs and iPRGCs remains unclear. 58. Although melanopsin is downregulated after light exposure, it could have contributed to iPRGC damage. As mentioned above, when melanopsin is activated by blue light, it produces reactive oxygen species, 41 to 43. Light-activated melanopsin may open transient photoreceptor potential calcium ion channels in the cell membrane, leading to an influx of calcium ions and further production of reactive oxygen species in the mitochondria. 58. Investigating the potential role of melanopsin in direct damage to iPRGCs from light exposure would be an interesting topic for future studies. We speculate that the lesions observed in the RGCs, which could include iPRGCs, may have had at least two effects. First, because the mitochondria in the axons and dendrites of these cells may help to meet the high energy demands of signal transmission. 24. The mitochondrial lesions may have disrupted signal transmission between iPRGCs, other retinal neurons, and various brain regions responsible for image and non-image forming responses. Second, the changes in the rough endoplasmic reticulum, coupled with a reduction in the number of free ribosomes, may explain the reduction in melanopsin staining that we saw, which is consistent with quantitative studies on the effects of light exposure on melanopsin expression. Because differ. NT types of iPRGCs are involved in image-forming and non-image-forming functions. 59.60. It would be interesting to investigate whether all types of iPRGCs are equally vulnerable to blue light. In summary, we found that exposure to blue light reduces melanopsin immunoreactivity in the iPRGCs of pigmented rats. We showed that, although continuous exposure to blue light at 1000 LX for 2 days does not cause RGC death, it damages the mitochondria in axons and dendrites of RGCs, likely including iPRGCs. This suggests possible disruption of signal transmission between iPRGCs and other retinal neurons, and between iPRGCs and different regions of the brain responsible for image and non-image forming responses. Future work could use immunoelectron microscopy to study iPRGC morphology after blue light exposure and investigate whether and to what extent melanopsin is involved in direct damage to iPRGCs from light exposure. Acknowledgements. The authors thank Mark Leonard for help with statistical graphics, statistical analyses, editing and English language usage. We also thank Natalia Sorinska, Maria Hanuszewska and Jacek Stork for their technical assistance. Supported by Grant Number Deck 2017-1, XN4 838 from the Polish National Science Centre. The project was co-supported by the Ministry of Science and Higher Education under the Regional Initiative of Excellence Programme for 2019 to 2022, Project Number 010, RID 2018-19. Amount of funding, PLN 12 million. Disclosure, N. Ziolkowska, none. M. Chmielewska-Kreminska, none. Awiniarska, none. W. Sienkiewicz, none. References. 1. Tosini G. Ferguson I. Tobota K. Effects of blue light on the circadian system and eye physiology. Review Mol Viz. 2016 2461-72. 2. International Commission on Non-Ionizing Radiation Protection. ICNIRP. Light-emitting diodes, LEDs. Implications for safety. Health Viz. 2020 118 549-561. Ratnayak K. Peyton JL. Megami, Godij NH, Gianfrido E, Karunarathni A. Blue light-triggered photochemistry and cytotoxicity of retinal. Cell signal. 2020-69. Crimsey, 547. 4. Grimsey, Wenzel A. Williams T. Roll P. Hafezi F. Remmer C. Rhodopsin-mediated blue light damage to the rat retina, effect of photoreversal of bleaching. Invest, Ophthalmal Viz Sci, 2001-42, 497-505. 5. Shang Y Wang G, Sliny DH, Yang C, Li L light-emitting diode-induced retinal damage and its wavelength dependency in vivo. Int J, Ophthalmal, 2017-10, Schumand, 1, 202. 6. Rohelex C, Schumann U, Ada M, Nels L, Funk RH, influence of blue light on photoreceptors in a live retinal explant system. Mol Viz, 2011, 17876-884. 7. G. Dane I, Villalpando Rodriguez G, and others retinal phototoxicity and the evaluation of the blue light hazard of a new solid-state lighting technology. SCI Rep. 2010, Sliny, 733. 8. Muller Ha, Sliny DH, retinal sensitivity to damage from short wavelength light. Nature. 1976, 260, 153-155. 9. Shang Y, Wang G's, Sliny D, Yang C, Li L White light-emitting diodes, LEDs at domestic lighting levels, and blue light reduces melanopsin expression, IOVs, or January 2022, or Vol. 6301, or Article 2610, retinal injury in a rat model. Environ Health Perspect, 2014 122, 269 to 276. 10. Jardine I, Boulengwes P, Chahari S, and others retinal damage induced by commercial light-emitting diodes, LEDs. Free Radic Biomed, 2015, 84, 373 to 384. 11. Geiger P, Barban M, Grimm C, Samadzija M. Blue, light-induced retinal lesions, intraretinal vascular leakage, and edema formation in the all-cone mouse retina. Cell Death Dis, 2015-6, 1985. 12. Iandiev I, Worm A, Holborn M, and others Muller cell response to blue light injury of the rat retina. Invest, Ophthalmal, Vis Sci. 2008-49, 3,559 to 3,567. 13. Kriegel A, Badugo M, Picard E, and others, light-induced retinal damage using different light sources, protocols and rat strains reveals lead phototoxicity. Neuroscience. 2016. 14 Versti 9, 296 to 307. 14. Kuz Y, Ogawa K, Suruma K, Shimazawa M, Hara H. Damage of photoreceptor-derived cells in culture induced by light emitting diode-derived blue light. CI Rep. 2014. 4, 5223. 15. Chu the 10th. Kumbalasiri T, Carlson SM, and others induction of photosensitivity by heterologous expression of melanopsin. Nature. 2005. 433, 745 to 749. 16. Gularad, Eka JL, Lol Gs, and others melanopsin cells are the principal conduits for rod cone input to non-image forming vision. Nature. 2008 453, 100, and 2 to 105. 17. Schmidt TM, Alum N, Chen S, and others, a role for melanopsin in alpha-retinal ganglion cells and contrast detection. Neuron, 2014-82, 781 to 788, 18. Galindo Romero C, Jimenez Lopez M, Garcia Ayuso D, and others number and spatial distribution of intrinsically photosensitive retinal ganglion cells in the adult albino rat. XP IRAS, 2013-108, 84 to 93. Denias J, Shen F, Goldblum D, and others cytoarchitecture of the retinal ganglion cells in the rat. Invest Ophthalmal Vis C, 2002, 43, 3, 587 to 594.20. Garcia Ayuso D, Galindo Romero C, D.E. Pier Domenico J, and others light-induced retinal degeneration causes a transient downregulation of melanopsin in the rat retina. XP IRES, 2017-161, 10 to 16. Benedetto M&M, Guido May, Contin Ma, non-visual photopigments effects of constant light-emitting diode light exposure on the inner retina of Wistar rats. Front neural, 2017, 840. 22. Bristow EA, Griffiths PG, Andrews RM, Johnson Ma, Turnbull DM. The distribution of mitochondrial activity in relation to optic nerve structure. Arch Ophthalmal, 2002 120 791 to 796, 23. Belenki Ma, Smaraski CA, Provencio I, Solas PJ, Picker GE melanopsin retinal ganglion cells receive bipolar and amacrine cell synapses. J-Comp neural, 2003 460 380 to 393.24. Osborne N, Lee GD, Mortarboys HJ, Jackson S. Light affects mitochondria to cause apoptosis to cultured cells, possible relevance to ganglion cell death in certain optic neuropathies. J. Neurochem, 2008, 105, 2013 to 2028, 25. Cheney, Soderberg PG. Lindstrom B. cytochrome oxidase activity in rat retina after exposure to 404 nanometers blue light. Kerr IRES. 2002 11825 to 831, 26. Garcia Ayuso D. Pierdomenico J.D., Vidal Sanz M., Villegas Perez MP. Retinal ganglion cell death as a late remodeling effect of photoreceptor degeneration. Int J. Mols. Survival of some photoreceptor cells in albino rats following long-term exposure to continuous light. Invest ophthalmal vis sci. 1976, 15 Chastifor to 70, 28. Hannibal J., Georg B. Fahren-Krug J. differential expression of melanopsin mRNA and protein in brown Norwegian rats. XPI, Res. 2013, 16, 55 to 63, 29. Mathies A., Engel L., Holthuis H., Woloshek T., Spazert R. daily profile in melanopsin transcripts depends on seasonal lighting conditions in the rat retina. J. Neuroendocrinol. 2007, 19, 952 to 957, 30. Sakamoto K., Liu C., Tosini G. Classical photoreceptors regulate melanopsin mRNA levels in the rat retina.
J. Neuroshi.
2000 and 424, 949, 693 to 9697, 31. Dreisig K., Łkowski, Blixt F., Wolffinge K. retinal cryosections, whole mounts, and hypertonic isolated vasculature preparations for immunohistochemical visualization of microvascular pericytes. J. Vis Exp., Davidson Rs., Nack Nami KC, Johnson GD, Davidson Rs., Mac Nami KC, Russell G., Goodwin D., Holborough EJ. Fading of immunofluorescence during microscopy, a study of the phenomenon and its remedy. J. Immunologic Meth. 1982-55 to 231 to 242, 33. Schindlin J., Arganda Carreras I, Fries E., Koenig V, Longair M., Peach T., and others Fiji, an open-source platform for biological image analysis. NatMeth. 2012-9 676 to 682, 34. Meijering E., Jacob M., Saria JCF, Steiner P., Hurling H., Answer M. Design and validation of a tool for neurite tracing and analysis in fluorescence microscopy images. Cytometry A. 2004 58, 167 to 176, 35, RCore Team. A language and environment for statistical computing. Vienna, Austria, R Foundation for Statistical Computing, 2021, 36. Wilcox R Introduction to Robust Estimation and Hypothesis Testing. Waltham, Ma, Academic Press. Waltham, Wilcox R Introduction to 7 and others, Cytoarchitecture of the Retinal Ganglion Cells in the Rat. Invest, Ophthalmal, Vis Sci. 2002, 43, 3, 587 to 594, 38. Hannibal J., Georg B., Henderson P., Fahren-Krug J. Light, and darkness regulate melanopsin in the retinal ganglion cells of the albino Wistar rat. J. Moll Neurosi. 2005, 27, 147 to 155, 39. Hannibal J., Georg B., Fahren-Krug J. melanopsin changes in neonatal albino rat independent of rods and cones. Neuroreport. 2007, 1881 to 85. 40. González Menendez I, Contreras F., Sanuda R., Provencio I, Garcia Fernandez J.M., postnatal development and functional adaptations of the melanopsin photoreceptive system in the albino mouse retina. Invest, Ophthalmal, Vis Sci. 2010-51, 2,840 to 4,847, 41. Tao J., Joe W., Jew the 10th. Mitochondria, as potential targets and initiators of the blue light hazard to the retina. Oxid Medcell Longev. 2019-2019 6,435,364. 42. Koyanagi M. Terakita A. Diversity of animal opsin-based pigments and their optogenetic potential. Biochem Biophys, Acta. 2014, 1837 710 to 716.
43.
Politini Mo. Moras M. Ramos Geronimo R. Castrucci AML. TRP channels, a missing bond in the entrainment mechanism of peripheral clocks throughout evolution. Temperature. 2015 to 522 to 534. 44. Dacey DM, Liao Hodge W, Peterson BB, and others melanopsin-expressing ganglion cells in primate retina signal color and irradiance, and project to Thel Yen. Nature. 2005, 433, 749 to 754. 45. Sonderrecker KB, Onyak Jr., Islam SW, Ross CL, Renner JM. Melanopsin ganglion cell outer retinal dendrites, morphologically distinct and asymmetrically distributed in the mouse retina. J. Comp Neural, 2017 525, 3,653 to 3,665. 46. Lucas R.J., Lowell G's, Allen A.E., Brown T.M., How Rod, Cone, and Melanopsin photoreceptors come together to enlighten the mammalian circadian clock. Prog, Brain Res. 2012 199, 1-18. 47. Won J., Jung H., who by, and others, acute photoreceptor degeneration downregulates melanopsin expression in adult rat retina. Neurosi Let. 2006 448-52, 48. Simo M., Lupi D., Pearson S.N., Butler J.N., Foster R.G., light-induced CFOs in melanopsin retinal ganglion cells of. Blue light reduces melanopsin expression. IOV's January 2022, or Vol. 63 or Number 1, Article 26 or 11. Young and aged rodless coneless, RADRDCL, mice. J. Nuroshi, 2007 and 7 to 3017-49. 2TC, Owens L.A., Anderson L., and others in a retinal photoreception independent of the visual retinoid cycle. Procnatal Acad Sci. USA, 2006 103 Pen, 426 to 10,431. 50. Núñez Alvarez C., Osborne N. Blue light exacerbates and red light counteracts negative insults to retinal ganglion cells in situ and R28 cells in vitro. Neurochem. Int. 2019 125 187 to 196. 51. Osborne N. Núñez Alvarez C. Del Olmo Aguado S. The effect of visual blue light on mitochondrial function associated with retinal ganglion cells XPI Res. 2014. 128, 8 to 14. 52. Nadal Nicholas FM, Madeira MH, Salinas Navarro M, and others transient downregulation of melanopsin expression after retrograde tracing or optic nerve injury in adult rats. Invest, Ophthalmol, Visai, 2015, a 56, 4,300, and 9 to 4,323. 50 sick. Sobrado Calves M, Vidal Sanz M, Agudo Barriuso M, long-term effect of optic nerve exotomy on the retinal ganglion cell layer. Invest, Ophthalmol, Visai, 2015 B56, 6,095 to 6,112. 54. Q, Ren C, Solas PJ, Pickard GE, so KF. The injury-resistant ability of melanopsin expressing intrinsically photosensitive retinal ganglion cells. Rev Neurose. 2015 284, 845 to 853. 55. Rovia G, Nadal Nicholas FM, Wang J, and others melanopsin-containing or non-melanopsin-containing retinal ganglion cells response to acute ocular hypertension with or without brain-derived neurotrophic factor neuroprotection. Invest, ophthalmol, vis sci. 2016-57, 6,652 to 6,661. 56. Garcia Ayuso D., Pierdomenico J.D., Esquiva G., and others, inherited photoreceptor degeneration, causes the death of melanopsin-positive retinal ganglion cells and increases their co-expression of BRN3A. Invest, ophthalmol, vis sci. 2015-56, 4,592 to 4,604. 57. Mark Ree, Jones B.W., Watt C.B., Vaughan D.K., Organisiak D.T. Extreme retinal remodeling triggered by light damage, implications for age-related macular degeneration. Mall Vis. 2008, 14, 782 to 806. 58. Sang A, Chang E, Lu H, and others, light-induced retinal ganglion cell damage in vivo involves dextrose 1, Mol Vis, 2011-17 134 to 143. 59. Reifler An, Chervenak Api, Dolikian Me, and others. The rat retina has 5 types of ganglion cell photoreceptors. XP Res, 2015 130-17 to 28.
60.
Hatta, intrinsically photosensitive retinal ganglion cells. Many subtypes.
Diverse functions.
Trends Neuroshi, 2011-34 572 to 580.